Hi, welcome to another episode of Catch This. My name is Trudy Ray. It is now a little more than a year since SARS-CoV-2 emerged and started causing infection in humans, and we already have several highly effective vaccines against this virus. Vaccine science can be incredibly complicated, and because of my previous research experience in this field, I was very skeptical about the promise of a SARS-CoV-2 vaccine this soon. But I was wrong, and I could not be happier about that. Two of the vaccines that are currently being used were developed by Pfizer, BioNTech, and Moderna and consist of a messenger RNA, mRNA, that encodes the full-length SARS-CoV-2 spike protein. When this vaccine is injected into a vaccine recipient, the mRNA would enter cells and be translated by the host protein synthesis machinery into the SARS-CoV-2 spike protein, which would then serve as an antigen to promote an immune response. For more details about how these vaccines work, I suggest my previous video linked here. Today, I want to talk in more detail about the composition of these vaccines. mRNA vaccines are non-infectious and do not integrate into the genome, meaning that there is no risk of infection or mutations caused by inserted vaccine sequences. Although these vaccines are the first of their kind to be licensed for widespread use, the concept is not new. Reports of the first successful translation of a foreign mRNA in animals were published in 1990. And this technology has been refined ever since. Progress in the field has been hampered because both scientists and investors were concerned that the inherent instability of RNA would prevent its use for delivery as a therapeutic or vaccine. Anybody that has ever worked with RNA in a lab knows how fickle it is and how frustrating it can be to handle it. However, research in the past 30 years has shown that the stability of RNA can be increased through various modifications and delivery methods. One way you can modify a vaccine mRNA molecule is by placing it between two RNA sequences that don't code for protein. These are known as non-coding regions or untranslated regions, shown here as UTRs. These UTRs stabilize the mRNA and optimize it for translation. The ends of the mRNA, the left end being known as the 5' end and the right end as the 3' end, can be further modified by adding what is known as a cap and a poly-A tail. The cap consists of a modified guanosine nucleotide followed by 3 phosphates, or G-PPP, and serves as a recognition signal for the cellular ribosome to bind and translate the mRNA. The poly-A tail is simply a string of adenosine nucleotides, denoted as AAA here. But there are many more than 3. This is just symbolic, and this further stabilizes the mRNA. Okay, so these are modifications that you can make to the RNA. But I also mentioned delivery methods. A common method for encapsulating and delivering the mRNA into cells is by encasing it in a cocoon of phospholipids. Phospholipids are a class of lipids that have a hydrophilic head containing a phosphate group and 2 hydrophobic tails derived from fatty acids. For example, the mRNA molecule in both the Pfizer and Moderna vaccines is encapsulated in a lipid nanoparticle, which protects the mRNA from degradation and ensures proper delivery into cells. This particle right here would fuse with our cell membrane and allow the inside, the RNA, to enter the cytoplasm of our cells. Addition of cholesterol molecules makes the nanoparticle more fluid and is thought to increase its ability to fuse with our cells' membranes. Polyethylene glycol, denoted as PEG here, does several things. It increases the potency of the vaccine particle by hiding it from the host immune system. It makes the nanoparticle more water-soluble because there are many hydrophobic parts here. And it slows down the degradation of the nanoparticle because it does degrade after a while. One of the reasons why SARS-CoV-2 mRNA vaccines could be produced so quickly is because all of this basic science was already in place at the start of the pandemic. And although the SARS-CoV-2 vaccines are the first mRNA vaccines to be authorized by the FDA for emergency use, several mRNA vaccines have undergone clinical trials in humans before for at least 4 infectious diseases: rabies, influenza, cytomegalovirus infection, and Zika virus infection. Another factor that helps speed up the process of SARS-CoV-2 vaccine production is that, luckily, scientists were able to extrapolate the insight gained from the study of other coronaviruses to SARS-CoV-2. Like the spike protein of other coronaviruses, the SARS-CoV-2 spike protein is highly immunogenic and is targeted by neutralizing antibodies, which bind viral antigens to inactivate the virus and prevent infection of new cells. The spike protein also mediates binding of the virus to the ACE2 host cell receptor via spike's receptor binding domain, and fusion of the viral particle with the host cell membrane via spike's fusion domain. However, to mediate this fusion, the SARS-CoV-2 spike protein undergoes a structural rearrangement from its pre-fusion conformation. By 2017, scientists at the Vaccine Research Center of the National Institute of Allergy and Infectious Diseases had already determined that the pre-fusion form of Middle East Respiratory Syndrome coronavirus, also known as MERS-CoV, is more immunogenic than its post-fusion form. So, they had spent several years engineering a mutation that locks the translated spike protein into its pre-fusion structure. When the SARS-CoV-2 genome sequence was published 1 year ago, scientists were able to compare it to the MERS-CoV sequence and identify the exact location where the pre-fusion stabilizing mutation had to be made. And luckily, it worked, meaning that the mutation in the SARS-CoV-2 spike mRNA sequence stabilized the spike protein in its pre-fusion conformation. Conventional vaccine strategies have repeatedly failed to yield vaccines against challenging viruses like HIV-1, herpes simplex virus, and respiratory syncytial virus, also known as RSV. However, recent advances in mRNA vaccine technology show promise in immunizing against some of these diseases. For example, RSV, a virus dear to my heart because I used to work on it at the CDC, poses a substantial public health threat due to its association with severe morbidity and mortality in infants and premature babies. Despite 60 years of continual efforts, we still don't have a licensed RSV vaccine. And this is in part because natural RSV infection does not induce a durable immune response. We do know that the RSV-F fusion protein, this one right here, is highly conserved and elicits broadly neutralizing antibodies. And recent studies have shown that, similar to the case of the SARS-CoV-2 spike protein, most neutralizing activity in human serum, that's antibody activity, is directed against the pre-fusion form of the RSV-F protein. This observation inspired scientists at Moderna to develop an RSV pre-fusion F protein mRNA vaccine, with phase 1 clinical trial data showing promising results. One of the exciting features of the mRNA vaccine platform is that it's not only applicable to preventing viral diseases, but it can also be used for treating cancer. Cancer mRNA vaccines would target tumor-associated antigens that are preferentially expressed in cancer cells. Vaccination remains one of the most effective public health measures for preventing and controlling infectious diseases. However, conventional vaccine approaches using live, attenuated, and inactivated virus vaccines are time-consuming and expensive. mRNA vaccines can be made more quickly and cost-effectively than conventional vaccines because you don't need to grow and/or repeatedly passage viruses in cell culture. But we wouldn't know any of this about mRNA vaccine technology without decades of prior studies. And this further highlights the importance of regularly funding basic research. Let scientists work on what interests them, and good things will happen, even if the research seems trivial at the time. My name is Trudy Ray. I'll see you next time on Catch This.